Hello everybody, I'm Tompo Biamungu Mayange from the Department of Animal Resources, and we investigated this current research with other researchers from the Department of Animal Industry Convergence at Kango National University, and we were supervised by Prof. Won from Daegu University. So today I will be presenting about the biodegradability estimation of polylactic acid resin via composting. As we know, plastic in everyday life have brought innovation and convenience to humans. But when discarded, they do not decompose for a long time, causing environmental pollution. So, therefore, the demand to develop biodegradable polymers have received a great attention and have been developed to protect our environment. So during this process, while microorganisms degrade biodegradable polymers as feedstock, they obtain energy to reproduce themselves and produce salt, carbon dioxide, and water as byproduct. Recently, many researchers conclude that composting condition enforces the biodegradation rate faster than that in environment. So finally, for the introduction, I can say that this particular study was conducted to introduce the composition of the biodegradation test apparatus required for measuring the biodegradability of biodegradable polymers and the variety of methods of using the composting process. Then to reach our goal, we use first we use the method ISO 148551, which is the method for the determination of the ultimate aerobic biodegradability of plastics based on organic compound under controlled composting condition by measuring of the amount of carbon dioxide evolved and the degree of disintegration of the plastic at the end of the experiment. And this figure 1 shows the schematic layout of composting and the CO2 scrambling based on this method. Then for the composting, we add ammonium chloride as per the polylactic acid resin weight in order to meet the carbon to nitrogen ratio, but we didn't add in the blank compost. Then we use also two type of biodegradable polymer resin made by polylactic acid and distinguished by their hardness, where we'll have with CTF and XD. Then for 100 days, CO2 evolution was measured by CO2 scrambling and back titration. Then for the degradation percentage, we use this formula where C1 is the cumulative CO2 amount from sample compost, C2 is from the blank compost, and E3 is the carbon amount from the initial sample weight. Then the result, the table 2 shows the testing vessels of polylactic acid resin after composting. Then after 100 days, the reactor will open and the photograph we took some photograph. Then we found out that TF and XD sample was were different compared to those initial conditions. And when we opened the reactor, the white slime, where you can see in the picture, spread out TF and the XD samples and attached firmly onto compost materials. Then TF and XD sample were very solid initially, but Their strength was quite tendered since the structure of sample was broken down during the degradation. Then the table 1 shows the preparation of the composting vessel, where we have the 4 treatments, the sample, the moisture content of the sample, and the total weight of the treatment. Then T2, table 2 shows the degradation of compost and sample in basic in weight basic, and we can the important information to get here is the degradation percentage, which was 68.3 in cellulose, followed by TF 54, and finally the 44 of XD. Then the last figure here shows the cumulative degradation rate of polylactic acid, the two sample and the control with time elapsed. And we can see after in 100 days, the change we found, we can see TF and XD tend to be different while they are from same polylactic acid sample. So there are many explanation can explain these two change here. We can say that maybe they were the sodium hydroxide we used was in low concentration, or we have leakage in the reactor. So to conclude, the polylactic acid resins degradability in nature could be determined under composting conditions before being produced. Then, based on weights between the initial and the final sample, cellulose, which was the positive control, followed by TF and XD, had been degraded from 60 to 60.8 to 44 percentage, and then 45.2, 54.2, 51.3, and 41.8 of cellulose, TF, and XD under CO2 analysis, which might be caused by troubleshooting of gas collection also. And the polylactic acid resin was readily degraded as cellulose. Then finally, the tenth of the sample was in order of cellulose, TF, and XD. Thank you for your time.